morning to you all students and learners. We are now looking into a course on immunology and immunoinformatics, wherein the first 4 units have been covered. The first unit was on overview of the immune system, which is having around 7 to 8 topics which we covered. The second unit was on immunoglobulins, about 4 to 5 topics. The third unit was on antigen antibody interactions, a sort of techniques and protocols, that also we covered about 5 to 6 topics. The fourth unit was on T cell and B cell development. Okay. So, afterwards now we are having the unit 5, topic 1 pertaining to the immunoinformatics. Okay. So, why the immunoinformatics? As I told you in the beginning, and myself Professor S. Krupanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, situated in a rural vicinity, Vadlamudi, district of Guntur, Andhra Pradesh, India. Okay. So, now in this immunoinformatics, why there is a requirement of immunoinformatics? Because it is so bewildering, That is, if you take up the immunoglobulins, as many stars in the sky, so many immunoglobulins who can produce, there is potential with us. So, that is what let us see now. With regard to the HLA, there is HLA class 1 and HLA class 2. Okay. In the HLA class 1, there is HLA class 1 contains A, B, C, E, F, G classes. And again HLA 2, class 2 contains DP, DQ, DR, DM and DO also. Okay. So, like this the several alleles, each one is having several alleles. All those details we see now. Okay. So, that is what here in this immunoinformatics unit, we got one topic on immunoinformatics. The second topic is on epitope mapping and epitope design. The third topic was on web-based tools for vaccine design. The fourth topic was on immunogenetics. Okay. So, these 4 will be dealing in unit 5. So, now let us go to the details here. So, there is a video that has kept on reverse vaccinology. Okay. So, what is vaccinology here? So, usually whenever a vaccine need to be designed, it takes nearly 14 to 15 years, because vaccine is not something new. It comes from the bug, that is, it comes from the same pathogen. So, the pathogen has to be cultivated, isolated, later it has to be processed for the what is that called attenuation, and once it is attenuated, it has to be processed for the vaccination. So, there is a big procedure involved. Okay. So, now instead of that, so this procedure takes good many number of years, about more than 10 years. In the reverse vaccinology now, we are having what is it called the gene sequences. Through the gene sequences, we can make the oligonucleotides, and through the oligonucleotide sequences, we can what is it called cultivate in plasmid, and through that we can express the antigen of our choice, epitope of our choice, and that we can prepare for the vaccine development. This is what the reverse vaccinology. So, in this process, within 2 years, we can make the vaccine. So, like this, a video, please go through this.
The computational reverse vaccinology project is after accelerating vaccine development using image processing and computer graphics tools that are available in cloud infrastructure. The traditional method of creating vaccine includes 3 steps: isolating the microbe, inactivating it in the lab, and then injecting it into microorganism. There are drawbacks of such approach. It cannot be used on all pathogens, may lead to side effects, getting the person sick, and it may be too time consuming. The new development is called reverse vaccinology, and it is to use the surface proteins that triggers immune systems for creation of the vaccines. The challenges includes that we have to deal with too many genes on the virus. Protein-to-protein interaction can be quite complex, and viruses may mutate too fast, and by the time the vaccine is created, it may not be viable anymore. The project proposal includes 3 steps. 1, narrowing down the variables, identifying the genes responsible for surface protein. 2, reducing the problem space, more specifically identifying target protein and using those as heuristic for viral protein function. And third, bounding the variations in problem space, predicting mutations of the viruses and using the mutated virus as a path for creating the vaccine. Although there are tools available and are used by scientists, but we found out that the tools we have from other fields of computer science, like image processing and computer graphics, can significantly accelerate vaccine development. The project will use data from COVID-19 and protein 3D infrastructure, and it's a collaboration between university and industry.
So just now we have seen the video wherein the cultivation of the pathogen is shown, and all types of the pathogens we cannot use for this type of the vaccine. Particularly for malarial parasite, there is no vaccine. It is better to go for the reverse vaccinology to have this type of the vaccine. Even for trypanosoma, several people in African country are suffering from the trypanosoma, sleeping sickness, and we can devise the vaccines through this reverse vaccinology. Okay, so now those things have been explained here. We go to the details of this topic now. So here the objectives for this immunoinformatics topic is to appreciate the computer-aided vaccine design. So earlier the immunoinformatics all deals with the computer applications, computer-aided vaccine design. The second one is to review the bewildering complexity of immune response components. Immune response components. Once there is immune response, okay, the B cell clonotypes and T cell receptors, okay, and again sequential epitopes and conformational epitopes. Okay, so like this, good many number of all these things will appear. So that's what here immune response components. After the response arises, these are all the components which we have to evaluate. Then to acquaint with web-based tools that are useful for immunoinformatics, and again to know the scope of immunoinformatics. Hereafter the scope lies with immunoinformatics. Now let us see the expected outcomes. The learners and the students of this program, of this course, are able to learn this type of aspects, like the wide array of immune complexity will be known. Wide array of immune complexity means the B cell clonotypes, T cell receptors, and again sequential epitopes and conformational epitopes, and again HLA class 1 types. Okay, each one is 10 to the power of 11, 10 to the power of 13, 10 to the power of 15, 10 to the power of 11, like this several. Okay, so that is the complexity we will be knowing now. And again the software tools to predict epitopes will be known, and the future potential of immunoinformatics will be appreciated. So in order to have the appreciation of the future potential, you should know the basics of immunology, which we dealt in the first 4 units. Okay. Now, why computational aided vaccine design? Is it needed, computational aided vaccine design? As I told you in the introduction, that the regular conventional process of making the vaccine, wherein the microbe need to be cultivated, and through that microbe cultivation we have to develop a sort of trials wherein convalescent sera we take. Through that sera we have to determine what are those antigens. The procedure is there. So instead of that, the computer-aided vaccine design. Now in the coronavirus, what are the spike proteins we are familiar now? The spike protein sequences already have come. So with that sequences, one can try the signature sequence of the spike protein of coronavirus can be tried to develop this type of the vaccine. Okay. So why computer-aided vaccine design means immune system is so complex. That's what we are going for, the computer-aided design. So here there is 10 to the power of 13 MSC class 1 haplotypes. You are all familiar what is MSC class 1. MSC is nothing but HLA, major histocompatibility complex or human leukocyte antigen. Both are look alike, Shiva Shankara like that. Okay, so this is present on chromosome 6 and they express here. Okay, and again there is MSC class 1 B is highly polymorphic. Okay, so that's what MSC class 1 is. Why here MSC class 1 is specified here? Because much of the vaccines are viral based vaccines. For viral based vaccines, MSC class 1 responds. And there is 10 to the power of 15 different T cell receptors. T cell receptors too many maximum among all what I am listing now. And then 10 to the power of 12 B cell clonotypes, so many B cells, so many clonotypes, so that this gives the immune potential. If everybody speaks of boosting the immune potential, boosting of the immune potential is only having B cell clonotypes, good many number of B cell clonotypes against the pathogen. Okay, that's what here. Then linear epitopes, that is sequential epitopes, 10 to the power of 11, and conformational epitopes, 10 to the power of 11. These numbers are far many compared to the DNA sequences. Now, key questions in vaccine design. So, let us know what is vaccine, what are the questions, which are the immune cells, immune system that are responding to this. So, which immune response should the vaccine elicit? Okay. So, let us see. T helper cell mediated immunity. That is what we have been speaking. Vaccine has been developed based upon the concept of the adaptive immunity. Innate immunity, adaptive immunity. Adaptive immunity is having the memory. So, once the vaccine is given, because we retain the memory of the vaccine epitope forever throughout our life, that is the purpose. So, for maintaining this memory, T cell mediation is required. That is what here T helper cell mediated immunity. So, that is what, which immune response should the vaccine elicit? It is the T helper cell mediated immunity, that is the first one. The second one is T cytotoxic cell mediated immunity. T cytotoxic cell mediated immunity is particularly interested for viral pathogens, viral vaccines. Then humoral immune response. Humoral immune response means in the adaptive immunity, the humoral immune response is immunoglobulins. Okay. And again, the cytokines are also involved here. So, these are the three we have to focus. So, let us see the linear B cell epitopes. So, linear B cell epitopes means they are all contiguous sequence of amino acids. Let us say these are all the sequences. This is from parasite, that is bacteria, Anaplasma marginale from cattle. And here there is sequential epitopes. Linear means sequential, one after the other. Okay. So, we can, so this is from IMTech, from what is it, Raghava, Professor Raghava from IMTech, from Chandigarh. They developed this website. Each one of you can go there and see the details. There is in India, Department of Biotechnology has given the opportunity to 2 or 3 centers. One is the IMTech Chandigarh, the second one is University of Pune, and again the IIT Delhi. So, there are supercomputing facilities. So, these 3 centers, our Indian scientists and our students are getting trained. Okay. So, it is as on par with any other international universities. Our Indian setup is developing through with the finance that is being provided by Department of Biotechnology, Government of India, for information of the B cell epitopes. So, now let us see the linear B cell epitopes. So, the definition, what is linearity here? So, epitopes formed by molecular interactions of series of hydrophilic amino acids. Hydrophilic, that is what you have to remember here. So, that means an amino acid like what is that called, glutamic acid, aspartic acid, they have got the minus charge, and again you can take lysine, and again you can take arginine, they are having the plus charge. That is what hydrophilic amino acids in the antigen. So, now prediction of linear B cell epitopes. So, linear B cell epitopes easy to predict, and they are not good immunogenic, linear one. Okay, prediction is based upon hydrophilicity. In the epitope there are amino acids. If these amino acids are having hydrophilicity, they can bind to the corresponding regions on the antigen. And again, flexibility and secondary structure of this antigen is also required. Now, let us see the prediction of conformational epitopes. Conformational epitopes are little bit difficult compared to the linear epitopes, because linear means in the antigen they are getting bound to the antibody in a linear fashion. Conformational means the antigen is always folded, protein is always folded, antigen is folded. So, different foldings will come towards the Fab region of the antigen. So, that is what here, much more difficult than linear B cell epitopes, based on similarity of the 3D structures of different antigens. So, antigen, as we see here, let us see the example. So, let the antigen be like this, let the antigen be like this. Okay. So, it is this and this are not sequential. in linear fashion. These are all two different conformational. So an antibody that is developed may be like this. Okay, so this region and this region constitute conformational epitopes. Okay. So hence 3D structures of antigens must be known. We should know the 3D structures. Now let us see there are several tools available for B-cell epitope prediction. Let us see one after the other. These are all the databases, and still there are plenty. For the sake of convenience of visibility, I reduced it here. So the first one is AB check. It does the errors in the sequences of the epitopes. The second one is AB pred, artificial neural network for B-cell epitopes. The third one is APG. Here again the structures and sequence alignments will be shown. Here antigen. Here again binding data, quantitative binding data for B-cell epitopes is shown, and BCPEP, that is B-cell epitope database. B-cell epitope database, one can look into here, and DNA plot. Here again VDJ. So VDJ is quite familiar, it has been taught to you in the second unit, and here is variable zone and diversity region and joining region, constant region. Several genes are there, several alleles are there for each of these things. That's what good many number of immunoglobulins one can make. There's potential. And again IEDB, that is epitope database, immunoepitope database. Again here also B-cell epitope database we can get. So these are all the various database tools. Get acquainted with this. We have to explore and navigate. Then only will be familiarized with these ImmunoInformatics tools. Now again the University of Pune, they have got, as I told you earlier, IIM Tech Pune and IIT Delhi. So they got a server. So they do the service also to the scientists and students like us. So here whatever the protein of your choice, let us say for coronavirus spike protein you want to develop epitopes, you wanted to see the epitope. If once the epitope is known, we can have the multi-epitope vaccine design. Okay. So now enter PDB, that is protein data bank ID. If there is no ID with you, if you have already downloaded, now that one browse and enter the PDB format. So once you enter there, so you'll be getting, please ask for, there is what is it called, epitope, that is conformational epitope. CEP means conformational epitope you are asking, not sequential epitopes. Ask for this. So like this it evaluates relating to your protein and gives you the epitopes to your email. So here we have to provide the email. Okay. So like that you can get the information. So there are institutes which are being supported by Government of India to help our students to do all these affairs. Okay. So usually this type of things are being done by computational biologists. But we are only the bioinformaticians, we are users of the softwares that have been developed by the computational biologists. Okay. Now let us see the T-cell epitopes. T-cell epitopes have major differences to that of the B-cell epitopes. There we have seen both linear and sequential, that is linear means sequential and also conformational. Here in the T-cell epitopes they are all linear. Okay. And here there is CD8 cells and also CD4 cells are there in both the cases. Now T-cell related immunological databases, and again that's what I told you, this type of information is enormous, like genomics, immunomics. Okay, so like this. So let us see there are several databases like allele, frequency, antigen, there's DB, MSc, database MSc, anthropology, HIV immunology, IEDB, immunoepitope database, and IMGT, immunogenetics. IMGT means immunogenetics. Immunogenetics and HLA, human leukocyte antigen. So like this, varieties of the databases are there even for this T-cell epitope prediction, T-cell epitope prediction. Now let us have this particular view for again to revise our past 4 units, what we covered in immunology and immunoinformatics. So in this, this is exogenous antigen processing. See, if you take the antigen, if a vaccine is also an antigen, a small antigen like epitope they give. Okay. So the epitope directly cannot go to the B cell. It has to be processed and it needs to be presented through MSC class 1 to the TH cell and again B cell. Then only the B cell gets undergo the development, differentiation, maturation and produce what is it called the immunoglobulins. Let us see now. So that's why what I wish to let you know is antigen processing and presentation is most important aspect. If it is not processed, it is not presented, no, T cell cannot understand, B cell cannot understand, no immune response can be elicited. Okay, now let us see. This is the globular protein, which is exogenous, outside the cell. So that is engulfed. But let us say all this is a dendritic cell or a macrophage. It is engulfed. Once it is engulfed as endosome, this endosome, the lysosome provides enzymes to the endosome. The lysosomal enzymes are all hydrolytic. They digest the antigen is nothing but it is a protein. It's proteolytic digestion takes place here. Okay. So meanwhile, while it is moving like this, in the endoplasmic reticulum, there is what is it called? MSC class 2 is being made, and that is being clipped, that is being protected by invariant chain. Okay. So like this it continues. Now endoplasmic reticulum is ready, it is coming out, and again from this endoplasmic reticulum, the MSC class 2 is coming out as endocytosis, exocytosis, and both the things are now released out of the cell. Once it comes out, it is being expressed, it is being presented to CD4 T helper cell. CD4 T helper cell. Now the CD4 T helper cell takes this information to this what is it called, B cell. So B cell produces immunoglobulins. So all these steps one after the other takes place. Again I will explain here. This is the globular protein. It is taken by the endosome. So this globular protein needs to be digested to produce into fragments called epitopes. So for that lysosomal enzymes will be useful. So it keeps happening here. Meanwhile, this MSC class 2 is being made from the endoplasmic reticulum. So this invariant chain, and once it is made, it is being released. So both it carries along with this. This is what the epitope, peptide of this protein antigen, and this is taken out and it is being presented. It is now, this is the peptide being presented to T helper cell. Okay. So that's what exogenous antigen processing and presentation. So this is endogenous antigen processing. So let us say here a protein has been taken out. So endogenous means our own cells. So sometimes what is happening, worn out cells are there, cancerous cells are there. Okay. So we are having an organ, and this organ we are maintaining for about 40 to 56 years. So there is wear and tear. So those type of the organ, the wear and tear proteins will be there. They will be taken in, and for that they will be digested and peptides are formed. Okay. So once the protein is taken, it undergoes digestion in the proteasome. Proteasome is a mill wherein the protein is being cleaved, digested. As a result, peptides are released, and these peptides go to the endoplasmic reticulum. This is called endoplasmic reticulum. Okay. So in the endoplasmic reticulum, they are going TAP1, TAP2, transporter aided protein. Okay. Once through the transporter aided protein, the peptides come inside, and once the peptides come, there is MSC class 1. Okay. To the MSC class 1, they get bound. Okay. Once they get bound, they get released to outside as MSC class 1 presenting. MSC class 1 is presenting the peptide of this protein that is taken. So this presentation goes to the cytotoxic T lymphocyte. So here there is MSC, as I told you, there is 10 to the power of 13 MSC class 1 are there. So many. So all that there is a requirement of immunoinformatic tools. See, protein, proteasome, TAP protein, MSC molecule, and again presenting out directly to the cytotoxic T lymphocyte. So now like this MSC class 1 epitope prediction. So MSC also is having the groove. MSC class 1, MSC class 2 are having the grooves. In the groove, the peptide has to be accommodated. Okay. So now let us take the pathogen. From the pathogen, protein comes. From the protein, peptide comes. Through the proteasome, we have to, what we have seen, the peptide library, for which computational tools are required. And again from the peptide goes to the this MSC molecule for presentation. So again MSC anchoring residues, there is good many number of MSCs are there. As I told you, this HLA-B is more than 2000 alleles, okay, like this. So for that also there is a computational tools are required. And again from there MSC peptide complex. This MSC peptide complex to form, we require computational tools. Ultimately, the epitope is being presented. So like this, even for MSC, the epitope to get anchored to the MSC, we had to predict through the computational tools. Okay. Now, in silico vaccine discovery, wherein a wide range of softwares are available now. A few I have listed here for B cell epitope, a few I have listed here for T cell epitope. So how do you choose the best? Which one is the best? There is big confusion for all beginners like us. Okay, we are not basically computational biologists, we are not studied bioinformatics, we are now going in the wave where there is a demand for the subject for the students. Okay, so how do you choose the best? So now prediction accuracy should be there. HLA allele population coverage. See, if you are working with any vaccine, so we have to take our Andhra people, what is the HLA haplotypes. So for that HLA haplotypes, where the epitope that you are designing, what is the type of the vaccine. So that's what you should know. Population coverage. Population coverage means HLA haplotypes in that population. Dravidian population, Caucasian population, that is African population. Okay, like that promiscuous epitope prediction, later T cell epitope hotspots, then conserved epitope prediction. So these are all the various steps with which we should be careful enough to have a wide range of softwares, which one is the best to select for our experimental purpose. So now this is another PEP-VAC. This is again another type of the software tools. It can predict promiscuous epitopes, can identify conserved epitopes, can predict epitope with proteasome cleavage site, and produces less number of epitopes, hence making lab experimentation easier. So that's what one can try with PEP-VAC. So here the URL also is given. Now please see here, this is an article wherein, so these many potential epitopes, how many? More than 1,75,000 for a peptide, for an antigen, let us say for example spike protein. Of this, there will be 2,000 predicted, potential to the predicted filters through the software tools, of which around 49 confirmed epitopes. From there, around 5 consensus epitopes, that is from potential to predicted to confirm to consensus epitopes. Ultimately, these 5 epitopes with which we are developing the vaccine. So that's what, please follow this. So let us see the future of this immunoinformatics from genomics to immunomics. Earlier, genomics and proteomics is there for the past 15-20 years. Now, immunomics is taking the lead. Establishment of immunome database, the complete set of epitopes in an organism. Okay, modeling of cytokine networks and in silico vaccine trials. So that is the future now. Let us see the summary of this topic on immunoinformatics. Okay, so the complexity of the immune system, that is MSC class 1 how many, and B cell clonotypes how many, and T cell receptors are how many, sequential epitopes are how many possible, and again the conformational epitopes are how many. We have given the list. That's what the complexity of the immune system with regard to the MSC HLA types and T cell receptors, B cell clonotypes, and linear conformational epitopes are elucidated. The immune responses that a vaccine elicits have been made clear. The software tools for the prediction of epitopes are shown. The server at the University of Pune for conformational epitopes to upload PDB file is shown. And again, the future prospects of immunome database is highlighted. Okay. So with this, let us go to the study questions for you to work at home. So why there is a need of informatics tools to be adopted in immunology? Distinguish between linear and conformational epitopes. Elaborate on the future scope of immunoinformatics. Write about MSC class 1 epitope prediction, and describe the funnel shape shown to screen the hits in the epitope representing the virulent factors. So with this, let us acknowledge sincerely the online resources and public domains for the use of the content to develop the teaching material through this presentation. Thank you one and all. So we have covered the immunoinformatics databases, B-cell database, and MSC database, and other things. T-cell database, please go through this. Thank you one and all. Thank you.